والفسيولوجي وبالباثولوجي. ان شاء الله بنعطيكم بريك 5 دقائق عشان ترتاحون. زين، بسم الله. اول شيء بنتكلم عن digestive system. تمام. في digestive system اذا جاء الشخص نفسه بدأ عملية الهضم اللي في الماوث. الفود will go through the mouth or the oral cavity to the pharynx. The pharynx, which part? The part which is behind, which is the posterior part of the mouth. So from this part, the food will go down through the esophagus. Okay. بعدين من esophagus to the stomach, the digestion will start, and also will continue to the small intestine. Then after that, through the ileum to the large intestine, after that to the anus. Nice. Okay, this is just general concept. Now we will talk about the gastrointestinal system. Well, we have two parts, the primary organs and the associated glands. The primary organs, where we have the oral cavity, first one, the esophagus, and the stomach, and the small and large intestine. The associated glands, where we have the pancreas, the liver, and the salivary glands. Okay, first of all, Hassan, why you said is that these glands are what? Associated with the gastrointestinal system. Because the pancreas and the liver and salivary glands secrete enzymes, okay? These enzymes help to digest what? The food in the stomach, for example, or in the oral cavity or the small intestine. But don't confuse, the liver does not secrete these enzymes to digestion, to secrete something that we will talk about it later. But we know is that there's something that's secreted from the salivary glands to the oral cavity. Okay, I know is that you all know it, which is the amylase. It's important to break down of the carbohydrate. We will talk about the others, where there's some enzymes that digest the small intestine also. Okay. Our part here, which is, we will start with the esophagus. The esophagus, as a histological part, we will talk about the epithelium. The lining of the esophagus is epithelium, but which kind of, what is the type of this epithelium? Okay, it is squamous epithelium. Squamous, that means the type of cells are flat. Okay, and also it is stratified. That means this flat cells will be more than one above each other. Okay, so it's not one lining, يعني مو بطبقة واحدة من نفس السكويمس epithelium. And it is non-keratinized. That means it is not like the skin cell epithelium, which is keratinized. Okay. The anatomical structure, we have to know where the esophagus starts and where it ends. Okay, it will start from the upper part, superiorly it will start from the upper esophageal sphincter here to the lower esophageal sphincter here. Okay, what we mean by sphincter? We mean by sphincter, this is the narrowing here that will close and open. So this will close and open to help to do a function. Okay, so the upper esophageal sphincter here to close, to prevent the airway to go to the esophagus. Okay, because the air shouldn't go to the esophagus. And the lower esophageal sphincter, why it is there? Because there are sometimes when there's digestion in the stomach, the acidity will increase. Okay, and we don't want the acid to go to the esophagus, so the sphincter will close while there is a digestion of the stomach. Nice. Now, there is also another structure, which is the arch of aorta, which is shown now here, is that there is a crossing of the arch of aorta and the esophagus. We have to know where the esophagus presses the diaphragm. Actually, it presses at the level of T10. Okay, what is T10? T10 يعني الفقرات. احنا أخذنا الفقرات. We will divide it to numbers and name and letters. Okay, so most of us know it, but if someone doesn't know it, is that we will divide it to C1, C2, C3 كده يعني الفقرات نفسهم. to T1, T2, T3, like that. Okay, so we have to know that the esophagus presses the diaphragm or presses the diaphragm at the level of T10. Nice. Okay, what is the importance of knowing the length of the esophagus? Actually, what is the length of esophagus? It will start from 10 here to here, so it is 25 centimeter. Okay. Why we should know is that the esophagus is 25 centimeter? Because when we sometimes use the esophagoscopy, okay, when we sometimes use it to detect if there's any abnormality in the esophagus from inside, we have to know how much deep we should put this esophagoscopy. Okay, so we will enter it from the mouth, المنظار نفسه, until we reach the esophagus. But until what? Until this amount of or this length. Okay, nice. Okay, why we use the esophagoscopy? We use it to detect is there any carcinoma, which is يعني سرطان, and is there any foreign bodies that stick there in the esophagus. Okay. Now we will talk about the stomach. Actually, the stomach we divide it as an area to several types or several areas. Okay, so the first one, which is the first one, here, which is the cardia. Then after that, the fundus, which is the upper part here. Okay, then we will find the body here, and the enterium here. Then the pylorus. Okay. We have to know the names because sometimes we call structural relation to the name. For example, here is the pylorus. Okay, let's see here in the next pictures that here is the pyloric sphincter here. There's another sphincter here, which is in the lower part of the stomach. Okay, the pyloric sphincter. It is the name of it as the origin of the pylorus here. Tamam. So what is the another thing that we have to know here is that the production or the releasing of a gland. So we have the mucus and we have the acid. We secrete both of them in the stomach, okay? But which one for what? For example, the mucus for what? It is for protecting. It will protect the lining of the stomach from the acid because the acid can damage the lining of the stomach. But the mucus will protect the lining from inside, so there would be no damage. Okay, nice. And so, as a logic concept, where the production of a mucus will be there, and where is the acid releasing will be. Okay, so of course the body will be there will be acid releasing, and also the fungus. The cardia and the pylorus, there would be a production of the mucus. Okay, because this part are the terminal parts. Okay, so if we go up superiorly, we don't want the acid to affect this area because there is a different lining here, different lining or different type of cells. Okay, and it will be affected by the acid. So the production of mucus is important to be here and important to be here, so the acid can damage the duodenum inferiorly. Okay, let us see the other lining here or the layers from the inner part. Okay, we will find the mucosa. Okay, then the submucosa. After that, the muscularis, which the muscle layers, which is we have 3 muscle layers for different movements. Okay, because in the stomach we digest the food by mechanically and chemically. Yes, chemically, by using the acid and by using the muscles in the mechanic movements. Okay, so each of this muscular layer have different function, different movement action. Okay. And there is another layer which is serosa, which is the thin layer, شفافة تكون, covering the stomach. Nice. Also, we shouldn't forget is that there is 2 sphincter. We mentioned the lower esophageal sphincter, and there is the pyloric sphincter. The curvature here, we have the greater curvature. You can see here the greater curvature. The lesser curvature is there. Okay. And there is also the lining of the stomach. يعني طبقة السtomach الداخلية تكون مكونة لنا what we call it روغة. Okay, what is روغة? روغة is that النتوءات اللي تكون in the inner layer of the stomach. Okay, and stomach is not like this flat. It would be like this. Okay, why? Because we sometimes when you eat a lot of food, your stomach will be more enlarged in size. Okay, and it can get restricted. Yes, or contract. Okay. Now the histology as a general of the intestine. Okay, we have the same layers that present in the stomach: the mucosa, the submucosa, the muscularis, the serosa. Okay, we have the adventitia. What is adventitia? The adventitia, it is term that called when the serosa is not covered by the interperitoneum. By the parietal peritoneum. What is the intraperitoneal? What is the retroperitoneum? Let us see here in the picture. You can see is that there is what we call it the parietal peritoneum here, right? This parietal peritoneum covered the abdominal organ. You can see this is the red line. This is the parietal peritoneum covered the abdominal organs. For example, here they are the spleen, the stomach. Okay, they are covered by the they are part of the interperitoneal organs because the parietal peritoneum are covering them. Okay, but for example, the kidney, the aorta, the inferior vena cava are they are retroperitoneal because they are behind it. Okay, nice. The histological part, why we know is that we have a muscularis and there is the other layers. Why we have to know them are the submucosa and the mucosa, because when there's an abnormality and there's damage to these layers, we can know is that is there an ulcer or we can call it erosion, or we can call it fissure. Okay, it depends which layer are damaged. Okay, when all the layers are damaged, we call it ulcer, but when the muscularis are intact, we call it erosion. But and the fissure is something different when there is the damage is sharp in shape. Yes, like what you are seeing here. Yes. So we will talk about the small intestine. It is 5 meters long, so of course it is longer than the esophagus, which is what we mentioned, 25. Don't forget centimeters. Yes, the small intestine have 3 parts: the duodenum and the jejunum and the ileum. Okay, so we classified it. There is some junction between each one of them. We know it by the characteristics. We can differentiate between the jejunum and the ileum and the duodenum by the characteristics. Let us see what is the difference between the jejunum and the ileum. Okay, they are the main difference. Okay, the jejunum and here is the ileum. Obviously, from watching them or seeing the picture of them, we can see is that, for example, the jejunum is more and wide. It is wider. Okay, than the ileum. Okay, for example, what is the next is that the blood vessels, there is change here. So for example, the blood vessels, we will classify it to the vesa recta and the arcades. Okay, the arcades and the vesa recta. The vesa recta, it is the terminals. You can see is that this is the vesa recta. We call it like this because it is the terminals. Okay, but the arterial arcades, which is here in the beginning, they are less in the jejunum, but they are more in the ileum. Okay, and what is about the vesa recta? The vesa recta is more longer in the jejunum, but it is shorter in the ileum. Okay, you can see here. Nice. Also, there are more vessels in the jejunum. That's why the jejunum are more dark and red. Okay, when you take both of them, you compare the color. The jejunum will be darker, the ileum will be lighter because there is less vessels. Nice. Also, the lymphoid tissue, both of them have lymphoid tissue, but there are more in the ileum, and there are what we call it Breyer's patch, which is part from the immune system. Okay, but which one is longer? I mean in length, which one is longer, the jejunum or the ileum? We can see here from the pictures that, of course, the ileum is longer than the jejunum. Nice. Quickly, let's talk about the large intestine. The large intestine, we have to know its part. When we end from the ileum, we finish the ileum now. We finish the ileum, so we will start with the large intestine. Here we, after we finish from the ileum, there is the cecum. Okay, the cecum here will go after it, the ascending colon here to the superiorly, then the transfer horizontally, then after that the descending colon. Okay, there is the appendix here, which is stuck with the cecum. Okay, it is apart from the immune system because there are antibodies there. Okay, the descending colon here finished. After that, we will continue with the sigmoid colon and the rectum. You can see is that we call each one depending on the position and the in which direction it go through. Okay, we have also to know how much it is long. Okay, or each part long, because when we enter the colonoscopy, for example, we enter it from here. When we enter the colonoscopy like this, we have to know if we enter it, are we in the sigmoid colon or are we in the left colic fissure, which is this part, the angle here and the angle here, or are we in the transfer colon? So we have to know which part long. All the large intestine is 1.5 meter, okay, long. Okay, we will talk about each part in the next slide, but here is the appendix here. You can see the appendix have, which is the pink color, that one, this one. It have different positions. Sometimes it's like this, sometimes it is like this, sometimes like this. The base is the same, okay? The base is the same from here, but the tip of the appendix different depending on the position, okay? How we can see is that what is the normal? All of them are normal depending on people, different from people to other, but the common one, which is 40.3 percentage, it is retrocecal, okay? Retrocecal position. Now each part, okay, we will start with the cecum. It's obviously 6 centimeter because it is small, but when we go to the ascending, it would be 50 centimeter. Let's compare it to the descending. Of course, the descending is longer, so it is 25. It is more than 15. Okay. The transfers is very long, actually. It is 50 centimeter. Okay, we will compare the transfer colon to the sigmoid because they are both long, but the sigmoid is less because it is 40 centimeter. Okay, the rectum is 12 and the anal canal is 4. Nice. Now, what is the second thing that's important about the large intestine? And the wide of each part, because the sigmoid is likely to be obstructed because it is this is the narrowest part of the large intestine. The cecum is the widest one because it is 6 centimeter, as I remember, almost, yes, and like this. The sigmoid is 4 centimeter. Okay, yes. Let's go to the next part. Okay, now we will compare the large intestine to the small intestine. Okay, so of course there is no villi in the largest intestine, as we saw previously in the small intestine. The large intestine are wider than the small intestine, and it is shorter in length. Okay, also there are other characteristics. For example, the histora, which is النتوءات هذه اللي تشوفونها زي كذا. The small intestine will be like this, for example, but the large intestine like this. Okay. Now, what is there also? There is the tinea coli. Tinea coli is this is a muscle that in the lung of the large intestine. So you will see it like this. It will go like this. Okay. This is what we call it longitudinal muscle, okay, which is called tinea coli. Also, there is the epiblock appendage, which is the fat, the fat here, the large intestine, we can see it.